Hi, everyone. My name is Jordyn Schuch. I am from Plymouth High School, and my mentor was Anthony Maxwell. And my project is on the effects of benzene exposure on fetal T cells. So infections are a leading cause of death for children under 5 years old, including tuberculosis, meningitis, malaria, and measles. So during gestation, the macrophages are formed as part of the fetal immune system as early as 5 weeks. And later in gestation, T cells and B cells are formed during pregnancy as special immune cells. So many things can impact development of the fetal immune system, which will subsequently lead to immunological complications later in life, such as sensitivity to infections. There are 2 types of stimuli that can impact development of the fetal immune system, including infectious stimuli and non-infectious stimuli. Both stimuli will lead to the activation of the maternal immune system. Maternal immune activation causes susceptibility to asthma, cancers, and autoimmune diseases. For this specific project, we are focusing on non-infectious stimuli. So air pollution is a major player for impacting fetal immune development. Because of increases in climate change and loose government regulation, more people will be exposed to unsafe levels of air pollution. One of these being the wildfires that happened in Canada. We saw that specifically in Detroit, there were high levels of, there was bad air quality, as we see here in Detroit and New York City. So there are thousands of different types of air pollutants. For this specific project, we're focusing on benzene. Benzene is an organic chemical compound and is one of the top 20 most produced chemicals in the world. It can be found in crude oil, gasoline, cigarette smoke, volcanoes, and forest fire emissions. So when the body comes into contact with benzene, it is metabolized in the liver. The liver will metabolize it into different metabolites, including benzene oxide, phenol, hydroquinone, and benzoquinones. For this specific project, we're using hydroquinone as a surrogate for benzene because it is safer to use. So the overarching hypothesis for the Moore Lab, or for several projects in the Moore Lab, is that maternal benzene exposure impairs development of the fetal immune system via signals from the placenta. This will lead to immunological complications in the offspring when they are born. So now we need to understand what the placenta is. It is a transient reproductive organ that is critical for several aspects of pregnancy. This includes waste removal, exchange of oxygen and carbon dioxide, maternal adaptations to pregnancy, and protection from infections and immune-related functions. It's also important to note that the placenta is mainly compromised of trophoblasts. So we also need to talk about T cells. There are 2 types. T cells are cells from the thymus that are part of the adaptive immune system that fight off infections. There are 2 broad types of T cells, CD8 and CD4. CD8 are responsible for killing abnormal or cancerous cells, and CD4 are responsible for actually orchestrating immune responses. For this project, I focus on CD4 T cells, and there are also different types of subsets of CD4 T cells. For this project, we're focusing on Th1, which are inflammatory, Th2, which are anti-inflammatory, and T regulatory, which regulate immune response. We also need to know about the AHR pathway. So several air pollutants, including benzene, can activate the AHR pathway. When the pathway is activated, it causes inflammation, activation of the immune response, and impacts on immune development. The standard for determining if this pathway is activated is CYP1A1. So the objective of our project was to find out or answer the question, do signals from trophoblasts exposed to benzene impact differentiation of T cells? The hypothesis was that benzene activates the AHR pathway in trophoblasts, which induces the activation of inflammatory pathways that leads to the release of soluble immune factors which impair T cell differentiation. So this is a method we used. We incubated 1 million Jordyn cells. Jordyn cells are human cancer CD4 positive T cells with 1 ml of corresponding media. Then we refreshed these cells with 500 microliters of corresponding media every 24 hours for 3 days. The groups we used, we had our RPMI no treatment with our control group. we added hydroquinone. We added 25 micromolar of hydroquinone into RPMI. We also had a group of conditioned media from wild-type SWAN71 cells, which are first trimester extravilous trophoblasts. We also had added hydroquinone into that conditioned media. And then we had another group called RHQ wild-type, which are SWAN71 cells that are exposed to hydroquinone for 24 hours and then are refreshed with clean media, which just means that the media doesn't contain any actual hydroquinone, but there are still signals from hydroquinone-derived trophoblasts. Also, a note that 25 micromolars of hydroquinone does not lead to SWAN71 cell toxicity or cell death. So these are the actual results that we found. The first question we wanted to answer was, do signals from trophoblasts impact T cell differentiation? So we first looked at CD4 and CD25. CD25 is a marker for Treg, which again regulates immune response. So if we compare our no treatment or our control group to our conditioned media wild-type SWAN71, we see that there is no change. However, when we add hydroquinone into the RPMI, we see that there is a shift towards double positive for CD4 and CD25. When we add hydroquinone into the conditioned media, we see that there is a shift towards single positive for CD25. When we actually remove the hydroquinone from the media, we only see a single positive shift towards CD4. We also looked at CXCR3, which is a Th1 marker, and again, Th1 is inflammatory. When we compare our control to our conditioned media, we see that there is no change. However, when we add hydroquinone into the RPMI, we see that there is a shift towards double positive for CD4 and CXCR3. When we add hydroquinone into the conditioned media, we see a single shift towards CXCR3. And when we remove the hydroquinone, we see a single shift towards CD4. The last marker we looked at was Th2. We looked at CXCR4, which is a marker for Th2, and Th2 is anti-inflammatory. So when we compare our no treatment group, our control group, to the conditioned media wild-type SWAN71, we see no change. When we actually add hydroquinone into the RPMI, we see a shift towards double positive for CD4 and CXCR4. When we add hydroquinone into the conditioned media for wild-type SWAN71, we see that there's a slight shift towards CXCR4, but there is little change. When we remove the hydroquinone, we see a slight shift towards double positive for CD4 and CXCR4. So we see that hydroquinone shifts T cells to Th1. If we look at RPMI 25, we see that there is a large shift towards Th1 when we compare it to our control group. So just to summarize question 1 and answer that question, when treating with hydroquinone or RHQ, we show that there is an increase in CD4. Hydroquinone itself is causing cells to shift to a double positive state for CD25, CXCR3, or CXCR4. SWAN71 conditioned media with hydroquinone is causing a shift to only single positive for CD25, CXCR3, or CXCR4. The second question we asked was, is AHR activated in Jordyn cells? So we used CYP1A1 to mark an activated AHR pathway. When we compare all these groups to our RPMI no treatment, we see an increase in CYP1A1 in SWAN71 no treatment conditioned media, SWAN71 hydroquinone conditioned media, and SWAN71 RHQ conditioned media. This just means that SWAN71 conditioned media and hydroquinone activate the AHR pathway in Jordyn cells. This could be responsible for the differentiation changes we saw earlier for question 1. So the overall conclusion, we know that benzene exposure increases CD4, Th1, and Treg expression in Jordyn cells. Hydroquinone and hydroquinone-induced trophoblast signals are shifting Jordyn cells to be more CD4 positive. SWAN71 cells that were treated with hydroquinone have a shift towards single positive for either CD25, CXCR3, or CXCR4 when hydroquinone is still present in the media. In all our treatments, the AHR pathway is activated, and this pathway could be responsible for the differentiation changes that we saw. So I'd just like to thank everyone at the lab. That presentation.